All right, let's dive right in. Have you ever paused to wonder how traits, the very essence of who we are, aren't just passed down through individual genes, but how those genes are intricately organized and, well, linked together on our chromosomes?
Mm.
Today, we're taking a deep dive into some truly fascinating insights from Essentials of Genetics, 10th edition, specifically exploring the incredible world of linkage and chromosome mapping.
Yeah, and what's truly remarkable here is how early, seemingly simple biological observations really paved the way for this profound understanding of how our genetic material is structured.
Right.
And maybe even more importantly, how new variations are generated. We'll trace the path scientists took to figure out exactly where genes are located.
And why that matters so much.
Exactly, why that knowledge is absolutely fundamental to everything from basic biology to, identifying critical disease risks.
So our mission today is to cut through the complexity, give you a clear understanding of what's most important in this field. We'll explore the foundational concepts of linkage, revisit those historical breakthroughs that completely reshaped our understanding, delve into the ingenious experimental methods used to map genes, and then, crucially, connect these discoveries to their powerful applications today in the modern genomic era.
So by the end, you should have a pretty firm grasp on this essential piece of the genetic puzzle.
Okay, so let's cast our minds back to the early days of genetics.
Right.
We know Mendel established the idea of independent assortment,
Right, gene sorting independently.
suggesting genes on different chromosomes sort independently. But the core concept we're tackling first is this. Eukaryotic chromosomes actually carry many genes, and their positions are fixed along the chromosome's length. Doesn't that fixed location kind of challenge Mendel's elegant idea of independent assortment?
It absolutely does. And this is where the genius of early geneticists, well, it truly shines. As far back as 1903, Walter Sutton, working with Theodore Boveri, started bridging that gap between cytology, the study of cells, and genetics.
Okay.
Sutton pointed out a really fundamental problem. There had to be far more unit factors, which we now call genes, than there were chromosomes in most organisms.
Makes sense logically.
Yeah, so logically, some genes had to be on the same chromosome. And pretty soon, scientists began seeing evidence that certain genes didn't just assort randomly. They seemed to be transmitted together as if they were joined or linked.
Okay, so linked genes.
Exactly. These linked genes are simply genes located on the same chromosome that tend to travel as a single unit during gamete formation.
Unlike genes on different chromosomes.
Right. Those follow independent assortment.
That makes sense. But if genes are linked, do they always stay together? Is it a complete package deal every single time? Because here's where it gets really interesting.
Well, that's the million-dollar question, isn't it? And the answer is no, not always perfectly. Nature has a kind of trick up its sleeve to ensure genetic diversity. During a specific phase of meiosis, when homologous chromosomes pair up and align, forming what we call a tetrad. Think of it like a bundle of 4 chromatids.
Okay.
A remarkable event can occur. Segments of these paired chromosomes can literally exchange places. We call this crossing over or recombination.
Like swapping pieces.
Pretty much. Imagine 2 strands of yarn wrapped around each other, then breaking and rejoining in a new combination. This process reshuffles the alleles between those homologous chromosomes, creating completely new combinations of genetic information.
And that's key for variation.
Absolutely crucial for generating genetic variation within a species. Now, it's worth noting that similar exchanges can happen between sister chromatids during mitosis.
Right.
But those don't create new allele combinations because sister chromatids are, well, initially identical copies.
Okay, so if we're trying to visualize this,
Yeah.
maybe an analogy helps.
Let's think about it conceptually, maybe like in some of those classic diagrams. If 2 genes are on different chromosomes,
Separate shelves.
right, like
Yeah.
2 separate books on different shelves, when you pick books for a new library, you can pick any combination.
Got it.
This means you'll produce 4 genetically different types of gametes, and in roughly equal proportions. That's independent assortment.
So a truly random mix.
Exactly. Now, picture 2 genes that are linked on the same chromosome and there's no crossing over.
Chapters in the same book.
Perfect analogy. Unless something drastic happens, those chapters always stay together. In this rare scenario of complete linkage, you'd only get 2 types of gametes. The original parental types, no mixing.
The non-crossovers.
Precisely, the parental or non-crossover gametes. Now, complete linkage is uncommon in nature, but it's a useful theoretical extreme to consider.
So what happens most of the time then, when linked genes do experience that reshuffling when crossing over occurs between them?
That's when things get really dynamic. If crossing over happens between two linked genes, it's like our book chapter is literally tearing and swapping sections with a homologous copy.
Okay.
This exchange involves 2 of the 4 chromatids in that tetrad. The outcome, you still get your original parental gametes,
The non-crossovers.
but now you also generate 2 new allele combinations. These are your recombinant or crossover gametes.
And the key insight.
The critical insight here is that the farther apart 2 linked genes are on a chromosome, the higher the probability that a crossover event will happen between them.
More distance, more chance of a swap.
Exactly, which means a higher proportion of recombinant gametes.
So if you're predicting what trace offspring will have, understanding this difference between complete linkage and linkage with crossing over,
Yeah.
it fundamentally changes your expectations.
It completely shifts the expected genetic ratios. With complete linkage, if you did a standard cross, you'd see a distinct F2 phenotypic ratio, a linkage ratio, maybe like 1.2.1.
Yeah.
But more importantly, if you do a test cross mating, a heterozygote with a double homozygous recessive, and the genes are completely linked, you'd only see a 1.1 ratio of parental phenotypes.
Which is very different from unlinked genes.
Totally different from the 4 phenotypes you'd get if they were unlinked. And interestingly, when linked genes are really far apart on the same chromosome or on different chromosomes, the percentage of recombinant gametes can approach 50%.
So they start acting like they're unlinked again.
Pretty much. Genetically, they behave as if they're assorting independently at that point. And this leads us right into one of the most pivotal moments in genetics history, thanks to Thomas H. Morgan and his brilliant undergraduate student, Alfred H. Sturtevant.
Working with fruit flies, right? Drosophila.
Yes, in the early 1910s, Morgan had noticed that X-linked genes, like those for yellow body color and white eyes, or white eyes and miniature wings, sometimes separated during crosses.
Even though they were clearly linked on the X chromosome.
Exactly, even though they were linked.
So he was faced with a real puzzle. Where was this separation coming from in linked genes? And crucially, why did its frequency vary so much depending on which genes he was looking at?
Morgan proposed that this separation was due to chiasmata, these visible points of overlap you can actually see between paired homologous chromosomes during meiosis.
Yeah, the physical crossing points.
He hypothesized these were the physical locations where genetic exchange or crossing over was happening. Then Sturtevant made the critical, almost revolutionary leap.
The big insight.
He realized that the frequency of crossing over between any 2 gene locations on a chromosome is directly proportional to the physical distance separating them.
So closer genes.
Less likely a crossover happens between them, meaning they have a lower recombination frequency. Farther apart, more likely.
Can you imagine Sturtevant's eureka moment? The story goes he compiled data from Morgan's crosses, stayed up all night.
And by morning, he had created the very first chromosomal map. It's astonishing. He took recombination frequencies like yellow body and white eyes showed 0.5% recombination, white eyes and miniature wings 34.5%, and yellow body and miniature wings 35.4%.
Okay.
He noticed that 0.5% plus 34.5% was very close to 35.4%.
So the order.
He deduced the gene order had to be yellow-white miniature. He then defined a map unit, or mu, also called a centimorgan, cm, as 1% recombination frequency.
So yellow to white is 0.5 map unit.
Exactly. And yellow to miniature, 35.4 map units. These distances are additive, letting you build a linear map. Now, it's important to grasp something here.
Okay.
Even if a single crossover happens in every single tetrad between 2 linked genes, you still only observe 50% recombination in the resulting gametes.
50% max.
That's the theoretical upper limit, 50% observed recombination, which means genes more than 50 map units apart appear to assort independently, even if they're on the same chromosome. And Sturtevant and Calvin Bridges later showed this wasn't just an X chromosome thing. Linkage and crossing over happen on autosomes too. Oh, and a fascinating side note. Drosophila males have no crossing over at all.
No crossing over in males. That must simplify things sometimes.
It actually does. Further genetic mapping, yeah.
Okay, mapping 2 genes is impressive, foundational. But what if you want to figure out the order of 3 or even more genes? That's where things get even more complex, right? And where multiple crossovers become like our ultimate genetic super sleuths.
Precisely. When you look at 3 or more linked genes, you introduce the possibility of double crossovers, or DCOs.
2 swaps instead of 1.
Right. These are situations where 2 separate exchange events occur between non-sister chromatids, but within that same tetrad structure. Now, their frequency is generally the product of the individual single crossover probabilities.
Meaning they're much rarer.
Much, much rarer, which means they'll show up in the lowest numbers among the offspring. But that rarity is exactly what makes them so incredibly valuable for mapping.
And to successfully map these, you need to meet, 3 essential criteria.
That's correct. First, the parent generating the crossover gametes, usually the F1 female in a standard test cross, must be heterozygous for all the gene loci you're mapping. Gotta have variation to see recombination.
Makes sense.
Second, the cross has to be set up so you can accurately figure out the genotypes of all the gametes just by looking at the phenotypes of the offspring.
The test cross helps there.
Exactly. Usually a test cross to a homozygous recessive individual. And third, you need enough offspring, a large enough sample size to ensure you actually recover all the different crossover classes, especially those very rare double crossovers.
Okay, so walk us through the logic. How do you actually use those rare DCO phenotypes to determine the exact gene sequence?
Okay, here's the kind of cool trick. You start by identifying the most frequent phenotypes in your offspring.
Those are the parentals, the non-crossovers.
Right. They reflect the original arrangement of alleles on the parental chromosomes.
Right.
Then you find the least frequent phenotypes.
The rare ones, the double crossovers.
Exactly. Now, by comparing these rare DCOs to the common non-crossovers, you can deduce which of the 3 genes is sitting in the middle.
How?
The gene that appears to have flipped its position in the DCO types relative to the non-crossover types must be the one situated in the middle. Think about it. A double crossover swaps the middle piece.
Ah, I see. The middle one changes partners, so to speak.
Precisely. Once you've definitively established that gene order, you can calculate the map distances. You do this by adding up the percentages of all the relevant single crossovers in a region plus any double crossovers.
Why add the doubles?
Because each DCO actually represents 2 single crossover events, one in each interval, so you count them towards both distances flanking the middle gene.
Got it. And this works for different organisms.
Yeah, this principle applies whether you're looking at X-linked genes in Drosophila like yellow, white, and echinus, or, say, autosomal genes in maize like brown midrib, virescent seedling, and purple aileron. The logic holds. Now, despite all this precision, mapping estimates can sometimes become a bit tricky, especially over longer chromosomal distances.
Why is that?
Well, it's largely because some crossover events simply aren't detected by looking at the final gametes. For instance, if an even number of crossovers occurs between 2 gene loci.
Like a double crossover.
Exactly, like a double crossover occurring between just those 2 genes, it can actually restore the original allele arrangement on the chromatid.
So it looks like no crossover happened.
Right. It becomes indistinguishable from a non-crossover event just by looking at those 2 genes. This leads to an underestimation of the true genetic distance.
It's like trying to measure a really long winding path with maybe too short a ruler. You might miss some of the twists and turns.
That's a great analogy. The farther apart 2 genes are, the greater the probability that these undetected multiple crossovers will occur between them.
Okay.
And this phenomenon brings us to something called interference. Interference, or I, is basically when a crossover event in one region of a chromosome actually reduces the likelihood of another crossover occurring nearby.
So one crossover interferes with another one close by?
Often, yes. We quantify this using something called the coefficient of coincidence, or C.
How do you calculate that?
You calculate C by dividing the observed number of double crossovers by the expected number of double crossovers based on single crossover frequencies.
Observed over expected.
Right. Then, interference I is simply 1 minus that coefficient of coincidence. So I equal 1C.
Okay, give me an example.
Sure. In that maze example, if the calculation shows a coefficient of coincidence, C, of, say, 0.804, then the interference I is 1 minus 0.804, which equals 0.196, meaning that about 19.6% fewer double crossovers occurred than we would have expected if the crossovers in the 2 regions were completely independent events. This positive interference, where I is greater than 0, is actually quite common in eukaryotes, especially for genes that are relatively close together. Now, for a long time, geneticists theorized about crossing over. It made sense, explained the data, but seeing is believing, right?
Absolutely. So how did they get the proof?
Well, in the 1930s, 2 sets of absolutely groundbreaking experiments provided the direct physical proof. Harriet Creighton and Barbara McClintock working with maize.
McClintock, a legend.
A true legend. And Kurt Stern working independently with Drosophila, they confirmed it.
So it wasn't just a theory based on ratios. They saw this physical exchange happening on the chromosomes themselves. That must have been incredible.
It was a monumental achievement. In the maize experiment, Creighton and McClintock were incredibly clever. They used unique physical markers on chromosome 9 that they could actually see under a microscope.
Visible markers. Like what?
There was a dense, stained knob at one end and a piece of another chromosome that had been translocated attached to the other end. Really distinct features.
Okay.
They then looked at these visible physical markers alongside genetic traits linked on that chromosome, things like kernel color and the starchy, waxy characteristic.
Yeah, what did they find?
They discovered that when genetic recombination occurred, when the traits recombined in the offspring, there was a precise corresponding physical exchange of these cytological markers between the homologous chromosomes.
Wow. Direct visual proof.
Direct, irrefutable evidence. Crossing over involves a literal, physical exchange of chromosomal segments. Stern found the same in fruit flies using different markers. It solidified everything. Now, shifting gears slightly, while meiotic crossing over happens between homologous chromosomes.
To make gametes, right?
Right. A similar physical exchange can actually occur between sister chromatids during mitosis in our regular body cells, or somatic cells.
Okay, what are those called?
These are called sister chromatid exchanges, or SCEs. But here's the key difference. Unlike meiotic crossing over, SCEs don't produce new allelic combinations.
Why not?
Because sister chromatids are, by definition, initially identical copies of each other made during DNA replication. Swapping identical pieces doesn't change the genetic information.
Okay, so no new trait combinations emerge from SCEs. But why are these harlequin chromosomes, I think I've heard that term, because of their staining patterns, why are they still significant?
They're incredibly significant clinically because their frequency acts as a really sensitive indicator of chromosome stability and potential damage.
How so?
Well, the frequency of SCEs dramatically increases when chromosomes are exposed to damaging agents, things like certain viruses, X-rays, UV light, or various chemical mutagens.
So they're like a cellular alarm bell for damage.
Kind of, yeah. And even more strikingly, SCEs are highly elevated in individuals with a rare condition called Bloom syndrome.
Bloom syndrome.
It's an autosomal recessive genetic disorder. It's caused by a mutation in the BLM gene on chromosome 15, which encodes a type of DNA helicase, an enzyme involved in unwinding DNA.
And the symptoms?
Patients with Bloom syndrome typically have growth delays, sensitivity to sunlight, immune deficiencies, and a very, very high predisposition to developing various types of tumors. Their chromosomes are known to be extremely fragile and unstable.
Displaying lots of these SCEs.
Exactly, an excessive number of SCEs. So studying SCEs provides crucial clinical insights into genome stability and disease.
Okay, so we've journeyed through the classic techniques, the historical breakthroughs. Amazing stuff. But what about today? With all the incredible advancements in DNA technology, how has that transformed gene mapping?
This is where the field has truly, well, exploded into the genomic era. Traditional recombination-based mapping, while absolutely foundational.
Got its limits.
It really did, especially in organisms like us, humans, where you simply can't do controlled crosses over multiple generations. It's just not feasible.
Right.
The advent of DNA markers completely revolutionized gene mapping. These markers are essentially short, identifiable segments of DNA. They have known sequences and locations, and they act like indispensable landmarks all along our chromosomes.
Landmarks on the DNA map.
Precisely. And we've seen several generations of these markers develop over time.
Like what?
Well, early examples included things called RFLPs, or restriction fragment length polymorphisms. These are variations in DNA sequences that change how specific restriction enzymes cut the DNA, creating unique length patterns you could track.
Okay.
Then came microsatellites. These are short, repetitive DNA sequences, like maybe CA repeated over and over, CNA repeats. The number of repeats varies between people, making them useful markers.
And more recently?
More recently, SNPs, single nucleotide polymorphisms, have become incredibly powerful.
Just a single letter change.
Exactly. A single base pair variation. And these occur millions of times throughout the human genome. Their sheer abundance makes them invaluable tools for large-scale studies, especially looking for associations with diseases.
And these DNA markers have had monumental real-world applications, haven't they?
Absolutely. Cystic fibrosis is one of the earliest and most impactful success stories. The CF gene was mapped to chromosome 7 using these very DNA markers back in the 80s.
A huge breakthrough.
Huge. Fast forward to, say, 2007, and using SNPs, scientists identified 24 distinct genomic locations linked to 7 common human diseases, things like type 1 and type 2 diabetes, Crohn's disease, hypertension, rheumatoid arthritis.
So they could pinpoint regions linked to susceptibility.
Exactly. This ability to pinpoint genetic regions associated with complex diseases has dramatically accelerated our understanding of disease susceptibility and, importantly, potential therapeutic targets.
And the Human Genome Project, of course, must have been a game changer here.
Oh, absolutely pivotal. It provided what we call sequence maps, the ultimate detail, down to the actual nucleotide sequences, and also physical maps, which detailed the distances in base pairs.
And this data is available.
All of this incredible data is now publicly available in databases, like the NCBI Genes and Disease website. It means this modern bioinformatic approach. Well, it has essentially rendered traditional linkage mapping, as Sturtevant conceived it, largely obsolete for species where we have the full genome sequence.
We just look it up in the sequence now.
In many cases, yes. Though the principles of recombination are still fundamental to understanding inheritance and variation, it has fundamentally reshaped our approach. Take a really complex disorder like autism spectrum disorder.
Right, known to be very complex genetically.
Extremely. It's known to involve potentially hundreds of different genes, alongside various non-genetic environmental factors. Now, mapping studies have identified many candidate genes.
And they can help estimate risk.
They can help estimate risk. For example, population studies suggest if one child has autism, there's roughly a 25% risk for a future child in that family, significantly higher than the general population.
But precise prediction is still hard.
Exactly. Precise prenatal diagnosis based solely on genetics is still incredibly challenging because of the sheer complexity and the multitude of genetic and environmental factors contributing. It really highlights both the immense power and the current limitations of our mapping techniques when dealing with highly complex multi-gene traits.
Wow. So from those initial careful observations of linked genes, watching how traits travel together, to the incredible precision of modern DNA mapping and genome sequences, this deep dive has really shown us how scientists have painstakingly, yet brilliantly, uncovered the fundamental blueprint of life's organization.
It's been quite a journey of discovery, and this journey, it really raises an important question for us all, I think. Considering the vast and still unfolding complexity of the human genome and the ongoing discovery of new genetic variations all the time, how do you think our understanding of linked genes and their implications will continue to evolve and deepen? Especially now, in this era of personalized medicine and revolutionary gene editing technologies like CRISPR.
That's a truly powerful thought, isn't it? The implications are just...
Well, enormous. Thank you for joining us on this deep dive into the fascinating world of linkage and chromosome mapping. We really hope you gained some incredible insights and maybe a few aha moments along the way.
Yeah, what was useful?
Keep exploring, keep learning, and keep diving deep with us.